Okay, welcome everybody. Let's get started. So today's workshop has actually been going since about 2019. And when I first started my job, I was out visiting farmers in the region. And one of the first things that was said to me was, Margaret, we need help with organic nitrogen management. And Daniel Geisler, who's part of our workshop, was also starting his job at the time, and we met during an orientation, and we chatted about it and submitted some grants to start doing work on organic nitrogen management, and we've been doing it ever since. And Patricia Lazicki, who's also part of our team, was working in Daniel's lab at the time. So the three of us began working on this. And Patricia and I gave the first version of this workshop in my office in 2019 to about 30 farmers. And since then, it's grown and expanded. We've continued to do a lot of research, and we've expanded to include many of our colleagues who have great depth and knowledge and experience on these subjects. And then we developed the 8712 publication. So it's been a great long-term project, and it continues to evolve every year. Every year we make changes to this workshop, and we continue to improve it and add to it based on research and add to it based on the feedback that we get from our workshop participants. Maria, can you drop the demographics poll in, the introductory poll? So if you don't mind taking a minute and completing this poll, it helps us gather information about who's here so that we can share that and make sure that we are reaching as many people as possible. Great, thank you. So we have several people who take the workshop annually, which is really wonderful. And I feel like they're part of our team too now. And I bring it up because we cover a lot in this workshop. And so as we go through the materials, if you're new to organic production, or if you're new to nitrogen management or new to farming, be careful. I just encourage you not to feel too overwhelmed. We do some summary activities and some chances to work with the material, and we try to highlight, we cover all of the basics while also getting into some of the nuance. So don't hesitate to ask questions along the way, and we hope to be able to steward everybody along as we go through this process of going through a holistic presentation of nitrogen management in organic production. So today's agenda, we'll start with the welcome and introduction, and then Daniel will share, provide us an introduction to nitrogen. And then Amélie Godin will talk about nitrogen and microbes. And then we'll talk about soil organic matter. And I will close with a activity on establishing nitrogen demand, and then we will end for the day. So here's our full team. So by the end of the third session, you'll have heard from all of these folks. Michael Kahn, everybody here is works for the University of California. Michael Kahn is an irrigation and water resources advisor. Amélie Godin is associate professor of agroecology at UC Davis. Daniel Geisler is a cooperative extension specialist in nutrient management at UC Davis. Patricia Lazicki is the vegetable crops advisor for Yolo, Solano, and Sacramento counties. And I'm Margaret Lloyd. I'm the organic agriculture and small farms advisor for Yolo, Solano, and Sacramento counties. And Joji Muramoto is the organic production specialist based out of Santa Cruz. Okay, the objectives of our workshop. We hope that by the end of our workshop, you will be able to have confidence in understanding the basics of nitrogen transformations and dynamics in organic systems. We also hope that you'll feel confident estimating nitrogen release from cover crops, amendments, and soil organic matter. using online tools and the 8712 document. And lastly, we hope that you will know how to use soil and tissue tests to monitor the nitrogen status. So the 8712 document is the companion to our workshop. And you can find this online. I've included it in the emails that I have sent. And I'm dropping it in the chat now. So we will be referring to this throughout the workshop. And it's a great place to prepare for each workshop. It's also a great place to review in between, or if you have questions. So the second half of that 8712 document has these worksheets that are intended to allow you to work with the information. And so today we'll be covering part 1, the crop and demand, and we'll also be covering the soil organic matter parts of that worksheet. By the end of our workshop, you'll be able to complete all sections of this worksheet and also able to complete the final budget. And we will briefly go over this at the end of the workshop to confirm how to do it. Okay, Daniel. Well, it's a real pleasure that we have all these great instructors as part of our workshop. So thank you, Daniel, for being a part of our team. And the floor is yours.
Mm-hmm. Thank you. Good afternoon, everybody. Great that you all could join us for this workshop. So in my presentation, I would like to give you a brief overview of nitrogen in the soil. And those of you who have taken classes on nitrogen, you know the nitrogen cycle in the soil can be quite complicated. There are a lot of things going on, but we don't need to know every single detail of the nitrogen cycle. So I will focus really on the important things to help you understand what is happening in the soil and what it means when we talk about nitrogen mineralization or immobilization. And I'd like to advance the slide, which doesn't really work. Is it still the first slide you see?
Yes.
Okay, so we can distinguish three major pools in the soil. We have organic nitrogen, we have ammonium and nitrate. And the organic nitrogen pool is by far the biggest pool. Most of the time, that's probably 90% or more of total nitrogen in the soil. Ammonium and nitrate are smaller pools, but they are very important because these are the two pools that are available to plants. So plants take up most of the nitrogen in the form of ammonium and nitrate. Ammonium is quickly converted in aerated and warm soils to nitrate by some microorganisms in the soil. So if you have a warm and moist soil, probably nitrate is the major form of nitrogen the plants are taking up. Now the organic nitrogen most of it is not directly available, but there are soil microorganisms that decompose the organic matter, or that organic nitrogen is part of the soil organic matter. And soil microorganisms, when they decompose organic matter, they may release some of the nitrogen if they don't need it themselves in the form of ammonium. So that's what we call nitrogen mineralization. It's basically the conversion of organic forms of nitrogen into a mineral form, which is ammonium. And so as ammonium, or if it's converted to nitrate, these two forms are plant available and can feed the plant. The microbes are actually part of the organic nitrogen pool. I'd like to show them separately because depending on how much nitrogen is in the material they decompose, they may need some of the ammonium, or they always need some of the ammonium themselves, but sometimes they may even take up ammonium and nitrate from the soil solution and reduce the availability to plants. And we will see examples later in a few minutes during this presentation. Then, of course, you all know nitrogen can be lost from the system. So, for example, ammonium can be converted to ammonia. Ammonia is a gas and can escape to the atmosphere. We call that volatilization. It's mainly a problem when urea or some manure that are high in ammonium are left on the soil surface. Then ammonia can escape. If the material is incorporated or watered in, then ammonia volatilization usually is very low from agricultural soils. The nitrate can also be lost. One way is by denitrification, and denitrification happens in waterlogged soils. So when there's not enough oxygen available, microorganisms can use nitrate instead of oxygen and convert it to either atmospheric nitrogen, which is N2, or to nitrous oxide. And in most soils, unless you have a very clay-rich soil and heavy rainfall, heavy irrigation, most situations, denitrification losses, if you calculate pounds per acre, are probably not that big. But nitrous oxide is a greenhouse gas, and so that's why denitrification is an environmental concern as well. But if we talk about losses, probably the main pathway of losing nitrogen from the system is through leaching. Nitrate is an anion. It has a negative charge, and clay minerals and soil organic matter, they may also have a predominantly negative charge, which means that they don't really interact with nitrate. And nitrate moves with the water. So if we have downward movement in the soil, let's say with a heavy sprinkle or furrow irrigation, or with heavy rainfall, then basically the nitrate or much of the nitrate just moves with the water. So if that water infiltrates below the root zone, it takes nitrate with it below the root zone. And that's at one point, depending on how deep the groundwater is, at one point it will reach the groundwater. But if it's below the root zone, it's basically lost for our crops. So nitrate leaching is definitely the biggest, in most situations, the biggest pathway of losing nitrogen from the system. So in this presentation, I will focus on what microorganisms do when they have organic material. They decompose it and may either mineralize or release ammonium, or they may also immobilize some nitrogen and use it for themselves. And because it is a microbial process, there are a lot of factors that can affect it. And I'd like to have a look at these factors first. So as an overview, soil temperature plays a role, soil moisture, then the quality of the organic source. We will have a look at the nitrogen content and the carbon to nitrogen ratio especially. And then also the crop management, especially when you use, when you have cover crops or organic amendments, how you manage them will determine how much is mineralized from these materials. But let's have a look at soil temperature first. This is a graph from one of our studies where we used undisturbed soil cores, and we kept them at different temperatures. In this graph on the left is 32% Fahrenheit, on the right is 82, and the vertical axis, that's just a relative scale. 77% Fahrenheit would be 100%. And you can see that nitrogen mineralization increases with temperature within this range, and it's actually an exponential increase. So the warmer it gets, the stronger the increase. And that's, in general, microorganisms are more active when it's warm. And that's the reason why we keep our food in the fridge, because otherwise it would spoil much faster. And spoilage is often or most commonly also done by microorganisms. So it's a very similar process. Of course, mineralization wouldn't increase indefinitely. Once we have a temperature of 110, 120 degrees Fahrenheit, it will level off. And if it gets warmer, then some of the microorganisms may not survive and their activity will decrease again. But within the soil temperatures we commonly observe here in California, we can say nitrogen mineralization increases exponentially with temperature. And here are just two examples of soil temperatures. The vertical axis is from 40 degrees Fahrenheit to 80 degrees Fahrenheit, and the horizontal axis is an entire year from January to December 31st. And these two curves are the soil temperature measured at the depth of 6 inches at CIMI stations. The black line is here at Davis, and the brown line is on the Central Coast in Salinas. And you can see when you first look at the black line for Davis, the lowest soil temperature in winter is around 45 degrees, and it reaches about 75 degrees in summer at the depth of 6 inches. On the coast, temperatures are not a bit less extreme, so soil temperature is warmer during the winter, but doesn't reach as high a temperature as here in the Central Valley. So it reaches a bit more than 70 degrees Fahrenheit. So these are the common soil temperatures we find here in California. If you move south in the Central Valley, the Davis curve would slightly be shifted upwards. If you go to Fresno or Bakersfield, then it will reach 80 degrees Fahrenheit or even a little bit higher. But these are the ranges of soil temperature you can observe, or we can observe here in the central, or here in California. Then soil moisture is also important. Again, this graph, the vertical axis is the relative nitrogen mineralization from 0 to 1, 1 being optimal, not affected by moisture. And from left to right is the water content, the volumetric water content. And for a silt loam, the field capacity would be at around 32% moisture. Field capacity is the moisture content the soil has 2 or 3 days after a heavy rainfall. So when the excess water has infiltrated and the water that is plant available is left. On the left here is the permanent wilting point, and that's the moisture content where below that plants can no longer take up enough water to stay alive. So they would start wilt and would die eventually. So really between the field capacity and the permanent wilting point, that's the water that's plant available. And we can see the black curve is nitrogen mineralization. It is optimal when moisture is optimal for plants. So at field capacity, microorganisms are happiest. And then nitrogen mineralization decreases with decreasing moisture content. It doesn't reach 0 at the permanent wilting point. So even when the soil is too dry for plants to grow, there are still plenty of microorganisms that may live in very small pores where plant roots are just too big, they cannot access that water, but the microorganisms in these pores are still pretty happy and they just continue with their business. But at one point, when the soil gets too dry, then microorganisms are also no longer active and they cannot survive. So they need water. And also here, an example or from you know that when you produce hay or grains, they need to be dried to a certain point because otherwise you cannot keep them. They would also deteriorate over time. So microorganisms, most microorganisms, and especially those in the soil, they need some moisture to survive. But they're happiest when the soil is moist and warm. So when we combine these two curves with climate data from California, so the temperature curve, the exponential increase with temperature, the change with soil moisture, we come up with a graph that looks somewhat like this. And in the next slide, I'll just show you a bigger picture of this graph. So here it's based on some assumptions. To create this graph, we assume that we have a summer crop that is irrigated, and we use climate data from Davis. So what we found is, and again, the y-axis or the vertical axis is nitrogen mineralization relative. 1 would be optimal, 0 would be no nitrogen mineralization, and from left to right is from January to January. And what you can see during the winter, nitrogen mineralization is really low in California soils, and that's mainly because of low temperature. The moisture content would be fine because we have winter rain, so most of the time during the winter the soil would be moist enough, but it's just too cool. Then in spring, when temperatures increase, nitrogen mineralization increases too. It reaches an optimum during the growing season because the soils are warm, they are irrigated. But when the crop is harvested, irrigation stops, the soil dries out, so nitrogen mineralization drops too. Sometimes in September, soils are really cooling down as well. So then we see this strong decrease in fall due to lack of water, due to lower temperatures. Sometimes in fall, we may see a little peak. That may be the first rain in fall when the soil suddenly gets moist. Temperature is still warm enough for microorganisms to be more active. But in general, nitrogen mineralization and microbial activity in general decrease in fall. So early fall, it's probably moisture limited, especially in annual crops when we stop irrigating. And then later in the fall, it is temperature limited. Then the quality of the organic source also plays a role, and 2 properties that are very important are the nitrogen content and the carbon to nitrogen ratio of the organic material microorganisms decompose. Here is a graph that is for residues to show you how nitrogen content affects decomposition and nitrogen mineralization. It's data from laboratory incubations that were done at 77 degrees Fahrenheit. And you can see from left to right weeks from 0 to 12. And on the y-axis, the percent of the total nitrogen in the residue that was released or mineralized from here starts at 0 up to 100%. So when we have a residue with about 3 to 5% nitrogen, that could be a legume cover crop, or it could also be a broccoli residue that's incorporated. You see that nitrogen is released or mineralized immediately after incorporation, and it continues, but it starts slowing down and reaches probably a maximum at around 60% of the total nitrogen in that residue that is being released. And the rest we would find either as undecomposed residue or as part of the microorganisms or already part of soil organic matter. When we have a residue with a lower nitrogen percent, let's say 2 to 3%, that could be a grass cover crop, or at 3% could be lettuce residue, or at 2% that would be roughly a tomato residue. You can see that at 3% we still see nitrogen mineralization, but slower and less of the total nitrogen is released. At 2% we actually see some immobilization, so a decrease in plant available nitrogen. But then after reaching a minimum, after a few weeks, microorganisms also start adding nitrogen to the system or increasing the plant available nitrogen pool. But the total amount released with these kind of residues is much lower than the total amount released with a residue that has a much higher nitrogen content. If you have residues with less than 2% nitrogen, that could be some cereal straw or corn stalks, we most likely see a decrease in available nitrogen at the beginning or immobilization, and then a slow mineralization later on. But depending on the carbon to nitrogen ratio, we may not reach the level we had at the beginning. So the microorganisms almost permanently immobilize that nitrogen. So why is that? Why does the carbon to nitrogen ratio, the nitrogen percent in a material play such an important role? The reason is that soil microorganisms, they decompose the residue or the organic material. That can also be an organic amendment, a compost, or even soil organic matter, so any organic material. The microorganisms, they need nitrogen and carbon as building blocks for their biomass. So they don't decompose organic material because they want to do a favor to the plants. They decompose it because they want to grow, they want to multiply, they want to use these nutrients and the energy for themselves. So nitrogen and other nutrients are used to build the biomass, carbon too, but carbon has also a second function. It is used as an energy source for microorganisms. So we see net mineralization or an increase in plant available nitrogen when there is excess nitrogen in the material the microorganisms decompose. So if there's so much nitrogen in there, they are kind of limited by the carbon that's there, and they just can't use all the nitrogen that's in the food source, and they release some of it in the form of ammonium, and that's what we call nitrogen mineralization. On the other hand, when microorganisms decompose something that has a lot of carbon and little nitrogen, let's say straw, then they have all that carbon, all that energy, all that carbon to build the biomass, but really not enough nitrogen to grow. And in that case, if there is ammonium or nitrate in soil solution, they may actually take it up and incorporate it into their own biomass. And that's what we call nitrogen immobilization. And nitrogen immobilization, it's not a loss of nitrogen. The nitrogen is still there in the system, but at least temporarily, it's no longer plant available because it's incorporated into the microbial biomass. So the microbes have to die first and be decomposed by other microbes before that nitrogen can become available again. But that's the reason why the carbon to nitrogen ratio is so important and helps us understand better how much we can expect, how much nitrogen we can expect from a cover crop or an amendment. And next week, Margaret and Patricia will go into much more detail or have many more examples about different amendments or cover crops. So as a rule of thumb, we can say that when the carbon to nitrogen ratio is less than 20, we may see net mineralization or an immediate increase in plant available nitrogen. If it's more than 30, it's very likely that the microorganisms may use some of the ammonium and nitrate from solution and so decrease the available nitrogen in the system. Then, of course, the availability of carbon and nitrogen is also important, or materials have different availabilities. Some of them are really easy to degrade by microbes. Others are much harder to decompose. Just an extreme example, when we look at broccoli residue, it has a carbon to nitrogen ratio of 10. That's about the same carbon to nitrogen as soil organic matter. But when we apply, let's say 200 pounds of broccoli residue, we can see that up to 130 or almost 140 pounds of nitrogen can be released within 100 days after incorporation. On the other hand, in the soil, in the top foot, we may have 3,500 pounds of nitrogen per acre in the form of soil organic matter, but it is really not well available because it had all the readily available components have already been decomposed. So even though there is much more soil organic matter in the soil or soil organic nitrogen, the decomposition is much slower, and it would not reach the level of the broccoli residue. So clearly the quality of the material plays a big role and determines how much can ultimately be available or released. Then management is also important. And with management, one of the main factors is incorporation. And this is a graph from Richard Smith. He did a study. He placed two materials, a pelleted material with 4% nitrogen, that's a 442 material for organic growers, and feather meal with 12% nitrogen. He put them in little bags and either buried them in the field after planting or left them on the surface. And here you can see the percent nitrogen that was released from 0 to 90% on top over a period of 60 days. And you can see both materials, when they were buried, they released much more nitrogen than when they were left on the soil surface. So that's one important factor. If you want the nitrogen to be available, it would be much, or you can get much more out of it if you incorporate it, or if you can apply it as a liquid with water, and you can water it in. But left on the surface, these are these two curves, much less nitrogen becomes available, mainly because, first of all, the material is not in contact with the soil, and if it's left on the surface, it may dry out. On the other hand, when it's in the soil, it is kept moist and it is decomposed much faster. So these are the main factors that affect nitrogen mineralization. It's temperature, moisture, the organic material itself, but then also management. And of course, if management, we could also mention irrigation. If you keep the soil moist with irrigation, then decomposition and nitrogen mineralization is faster. So just to summarize the two main points, nitrogen mineralization or release of nitrogen from organic material is a microbial process, and it turns unavailable nitrogen into nitrogen that's plant available. And environmental factors, temperature, moisture, then the property of the organic material and the management all affect how much nitrogen is mineralized. Okay, that's my presentation. I hope you have some time for questions.
Great, thank you, Daniel. Okay, let's review the questions. I'm going to start with the questions in the Q&A. So if you put your questions in the chat, please move them over to the Q&A. And if you also want to raise your hand and ask a question, we'll keep an eye on the screen as well. Okay, so the first question in the Q&A is, how does heavy clay soil affect denitrification?
So heavy clay soil has a much higher water holding capacity, so it remains moist or can even be anaerobic for a longer time after any irrigation or a heavy rainfall. And so it is much more likely that you have anaerobic conditions in the soil or lack of oxygen. So then in a heavy clay soil, the risk of denitrification is higher than in a sandy soil that drains very quickly.
Thank you. Patricia, did you have anything to add? I saw a little note there that you were going to chime in. Feel free to.
Oh, no, not at all. Just we had a couple of denitrification questions. I answered some of them in the chat.
Thank you. Okay. What would be a general estimate in percent of N being lost to the atmosphere after conventional fertilization? Meaning, what is the relative efficacy of a given fertilization event?
So in conventional system, it really depends on the management. There are studies that have shown that, for example, if you apply urea to the soil surface, you don't incorporate it, you can lose as much as 30%. That's probably the highest amount. If you incorporate it, it may be very low, or it may not even be measurable. there's also in organic system, let's say if you apply a manure and leave it on the soil surface and it has a high ammonium concentration, you may also lose some ammonium, and or I mean you can even smell it when you have a slurry or an animal manure. You can smell the ammonia escaping. I'm not sure in percent how much can be lost, but it can be a considerable loss, especially when it's warm and windy. Then quite a bit of the ammonia of the ammonium is converted to ammonia, and the ammonia can escape. So in order to reduce ammonia losses, it is important for not to apply it when it's hot and windy, and incorporate it as soon as you can. Or in conventional agriculture, if urea is surface applied, to water it in. Just an inch of water or half an inch of water moves the material into the soil, and there the ammonium will interact with the soil, and it's not converted anymore to ammonia, or at a much lower rate.
Thank you. There are a couple questions about how deep are you referring to when you say incorporation, and also about Richard Smith's work. How deep was that?
I can't remember exactly, but it was probably 3 inches or so. And incorporation, it really, it depends a bit, but it doesn't have to be deep incorporated. But let's say probably if you the top half inch or inch of the soil will dry out, depending on your irrigation system. So if you can incorporate it a few inches into the soil, that would be ideal. But as long as it's basically, let's say, as long as it's incorporated into the soil that remains moist during the growing season, then you're fine. If it's very shallow, half an inch or so, and that soil dries out very quickly, then it's not that effective. But as long as the material or whatever you incorporate is in the soil that remains moist during the season or after application, then that's a good depth.
Great, thank you. I'm looking through the questions. A couple of them about microbes we'll save for after Amelie's presentation. And then there's a question about the ideal volumetric moisture content % range for sandy loam and loamy soils. Ahmad, I'm not sure what you're referring to, like for a specific purpose.
So ideally, I don't have the values in my head for every soil, but probably when you Google it, you will find what is for different soil textures, what's the field capacity and the permanent wilting point. And really between these two moisture contents, that's the water that's plant available. Of course, when you irrigate, you don't want to wait until the permanent wilting point is reached, because if you approach that, the plants are already, or they need much more energy to get all the water. So ideally, you want to keep the moisture content somewhere near the field capacity. And the more sand you have, the lower that soil moisture is, and the more clay you have, it is somewhat higher. But you can find these values online when you can Google it, and there are many documents highlighting the differences based on soil texture.
Great, thank you. Is nitrogen use efficiency affected by the rate of organic nitrogen mineralization?
It depends a bit how you define nitrogen use efficiency. It is relatively easy to determine nitrogen use efficiency in conventional systems, because you measure how much is applied and then how much more can plants take up compared to a control. In organic systems, of course, it depends on the nitrogen mineralization, because if you have a material that doesn't release a lot of nitrogen, then if you compare how much the plants take up if you compare that to the total amount you applied that year, then your nitrogen use efficiency can be much lower. But of course, the rest is not lost. It's still in the soil. It is converted to soil organic matter, and it will contribute to the crop growth in during the next years or for many years to come. So in organic systems, you really have to clearly define what you mean by nitrogen use efficiency. But if you do the same calculation as in conventional farms, you may be surprised how low it is, but it's not necessarily a bad thing. It's just less of the nitrogen becomes immediately available to the crop that follows your application. But that also means more is left in the soil in the organic form and contributes to soil fertility over the next few years.
Great. Thank you so much, Daniel. We'll take some of the rest of the questions offline. And Amelie, if you want to go ahead and share your screen.
All right. So glad to be here today. We're now actually going to build on what Daniel said and presented today to understand a little bit more in depth some of the microbial, but also soil food web drivers of nitrogen cycling and availability. Feel free to add your questions to the Q&A and then we can circle back to them toward the end of the presentation. So our roadmap for the next few minutes here and our main objective is really to better understand how soil life at the micro scale really regulates nitrogen availability. So we're going to start talking about who is there, microbe as part of that soil food web. And then what do they do? How do they process and cycle nitrogen? And then we'll dive into nitrogen management and microbial community, how some management decision impact microbes in the soil, but also at the soil-plant interface that we call the rhizosphere. And then what does that mean for soil fertility management and kind of bringing it back to how can this knowledge can help you take the pulse of what's going on in your soils. So we focus really when we think about nitrogen management on the outcome above ground, but as Daniel pointed out, it actually relies on below ground processes and really the plant teaming up with microbe in the soil. We often think of microbial process and soil food web as occurring in the bulk soil, so all across the bed, all across the field. But what I'd like to bring to the forefront today is that this zone where the root communicate with the soil and the microbe is the hot spot, where we have high levels of communications between plants and microbes and nutrient exchange. So this is where a lot happens. And in that zone, really, a teaspoon of soil can contain between 1 million to 1 billion bacteria and about 10,000 different species. And I'll get back to that in a minute. But the plant is really in close contact with the soil food web. The way I think of the soil food web, it's like the guts in our stomach. That are full of microbes that secrete enzymes and that process our nutritious food into some elements for us to grow. Same idea with the microbes living in the soil. They're really the guts of the soils, and building up that soil food web allows for more optimum nitrogen flow. Oh, sorry. So there's a lot of organisms in the soil, and they're all important. We can have, as you see here, some bridal-tail and spring-tail organisms. We go down to bacteria. This is a rhizobium, nitrogen-fixing bacteria in a nodule of a clover root. This is a predatory mite. It's a mite actually eating another organism. Here we have ectomycorrhizal fungi that are growing outside of the roots and that are stained in yellow. We've got earthworm, nematodes, mycorrhizal fungi inside the plant root, etc., including those guys here that are phage. And we know very little about them, but they're actually very important in nitrogen cycling because they're going to eat bacteria and eat fungi and be very, very important into cycling this nitrogen. So we've got a lot going on and a lot going on that goes very fast. And it really goes beyond microbes. What you see here on the left side is a soil that has microbe but doesn't have any fauna. And on the right side of your screen, this is a soil that has fauna. So it has more than bacteria and fungi. It has earthworms and nematodes. And so what we see is that all those organisms are actually very important into degrading and turning over the organic matter into some forms that is bioavailable to plants. We've got detritus feeders, bacterivores, fungivores, and a whole bunch of predators that feed on each other. Now, this is kind of what we see here. Organic nitrogen comes into the soil through organic matter, and it's being processed by a large web of organisms that are going to regulate the end cycling and availability and retention. So yes, microbes are important, but microbes are just the start of that nitrogen cycle in the soils. They're going to be eaten by meso and microfauna, and that nitrogen is going to transit through the soil ecosystems. So managing the nitrogen cycle means managing the soil food web, nitrogen mineralization that results from the soil biota feeding. So organisms feeding on each other is really what moves nitrogen through the soil food web. Soil biota, including microbes, are also important temporal storage of nitrogen. They have a lot of nitrogen in their body and carbon in their body that Daniel already mentioned. And they're the main component and the main organism for nitrogen transformation. They also drive nitrogen losses. The beginning of that food chain here is really the fungi and the bacteria. And I just want to point your attention of the frontier of science where we're at. If we take a soil and we sequence and we try to enumerate every single microbe that is there, right now our power is about 10%. We know about 10% of the organisms that are in soils. So it's pretty much a black box. When it comes to earthworm, about 23% of the earthworms we have in our soils are actually being characterized. So it's important to take into account when we think of managing microbes, adding inoculum, all of those things to steer microbial community. 90% of who's under our feet remains unknown. Now, what do they do? And I want to start here with some more typical end cycling on conventional farm, where you have most of the nitrogen coming in the form of fertilizer, a large pool of nitrate in the soil. When we think about organic system, it's mostly driven by organic matter input that enter the soil organic matter pool and the microbial pool, and most of the storage of nitrogen happen at this scale rather than in the form that is easily leachable. And I'm mentioning this because when we talk about nitrogen use efficiency, referring to your earlier questions, we've got a loop of nutrients here that are transiting between the microbe and the soil organic matter. And so of your cover crop, 15% is probably going to make it into nitrate and ammonium. The rest is going to be in those microbial carcasses, in that soil food web, and in that soil organic matter. So you're investing into that bank of nutrient for the future. Now, although all the food web is important in cycling microbe, when we think of mineralization and the first step of the nitrogen cycle, fungi and bacteria are some of the most important. So let's take back some of Danielle's nitrogen cycle here. I wanted to share with you a little bit more about the specific microbes and what is it that they do. So we've got some organic nitrogen that comes in through forms of residues and or compost. We can also have nitrogen fixation happening here. So those can be free living bacteria in the soil or bacteria that are associated with the plant. The most well-known is Rhizobium that hangs out with legumes in nodules. But there's a lot of different free living bacteria in the soil that are not in a nodule, that are not associated with a plant. And we thought for a very long time that those free living bacteria were not so important. But it turns out that there's 5 to 10% of the nitrogen that is unaccounted for if we look at an overall nitrogen balance in the field. And we're increasingly thinking that some of those free living bacteria are very important actors in the nitrogen cycle. Now we can know who they are because they have a specific gene in them, in those bacteria. And so we can look at who has this gene and how many copies of that gene is in the soil. And that gives us an idea about how much nitrogen fixation we might see. In terms of that first step of transformation from organic N to ammonium, which is mineralization, it's mostly done by what we call ammonifying bacteria. They include Bacillus, Streptomycetes, for instance. And here it's difficult to count them and to know especially who they are because there are many of them doing that job. And so there's not like a single gene that we can look at. And so it's a little bit more complex to see which bacteria is actually involved in that first step. The next step, which is nitrification, so it's ammonium being transformed into nitrate, is more characterized. And it involved any bacteria that starts with the name nitro something. Which means that the bacteria get their energy from this nitrogen compound that they will oxidize. So they're taking ammonium, they're ammonia oxidizing bacteria to get to nitrite, and then nitrite oxidizing bacteria to get to nitrate. So here we've got the Nitrosomonas and the Nitrobacter. There's other type of bacteria that we've all named nitro something. And those ones we know more who they are and what they do because they have this specific gene here that we can look at. We can look at how much of that gene is in the soil and kind of give us an idea about how much of those bacteria are in. And then finally, denitrification. Those are mostly those Pseudomonas. There's 3 genes that we know of that indicate that these bacteria might be denitrifying, but it's a very complex step as well. So a lot of different types of bacteria and their own genes are gonna, and their need for different nitrogen sources to grow and reproduce is gonna dictate their the step they participate in within the nitrogen cycle. Now, fungi are also very important. I guess one of the most well-known is mycorrhizae, for instance. Mycorrhizae are fungi that team up with the plant, that infect the plant roots or live around the plant roots and help the plant capture nitrogen, water, and phosphorus farther away from the plant. So we know that they help the plant with uptake of nitrogen, but also water. But we now know that some of those hyphae that are the structure of the fungi in the soil are also also have an impact into mineralization. So they also help break down this ammonification first step of going from organic nitrogen to ammonium. And then finally, something that we've discovered not long ago, and here you can see on that video, this is the little mycorrhizae hyphae. So that's a fungi. And they create what we call highways of moisture all around the hyphae here. And that provides some high-speed ways for bacteria to travel through. So they actually team up, where the fungi and the bacteria coexist, and the fungi is facilitating bacteria traveling through by providing those hydrochannels around the hyphae. So something kind of cool that is happening as well. And then in those fungi has a storage of organic nitrogen, as well as very important for water movement, aeration, microaggregate formation that has an impact, as we've seen previously, on soil moisture and potentially soil temperature as well. Now, Danielle mentioned that carbon and nitrogen is important, that the relative abundance of those two is important. To go a little bit more in detail, really, the microbes, whether fungi or bacteria, need carbon for their energy source. Okay, they use carbon and other organic forms like glucose, for instance. They're the main component of their cell wall. And now they do like us, they undergo respiration. And so some of that carbon is lost back into the atmosphere. They also use nitrogen that is used for them to produce protein, to produce their DNA. So very important part of who they are. Now, to grow, a microbe are going to need 8 units of carbon for each unit of nitrogen. That's what it needs for a balanced growth. Now, it's going to lose some via respiration. Right? So all of a sudden, we need 24 units of carbon per unit of nitrogen. And this is where these 24:1 or 20:1 comes from. The ideal carbon to nitrogen ratio is going to be based on the ideal microbial diet. And so if you have a carbon to nitrogen ratio in your residues that is more than 24:1, the microbes are going to need to find that nitrogen somewhere to sustain their growth. If it's less and scavenge nitrogen and therefore lead to immobilization. But if it's less than 24:1, there will be a rapid cycling of that nitrogen. So all this comes from microbial physiology and their own need for carbon and nitrogen, and also based on how much they need for respiration. So to recap and build a little bit on what Danielle mentioned, when you add a residue here, your microbial activity is going to increase. Okay, this is yummy food for your microbes. It's coming in, they're getting all jazzed up. Microbial activity increase if moisture and temperature is adequate. At the same time, the carbon to nitrogen ratio of your residue is going to decrease. And nitrogen available to the plant is going to increase as it's being released from the residue. This is if we have a carbon to nitrogen ratio of about 11, so a lower level of close to the microbial diet. Now, if you have a carbon to nitrogen ratio that is really high, like a wheat straw, for instance, instead of being here, you're here, the microbial activity is going to rapidly ramp up, but the microbes are going to need to scavenge for nitrogen to process that carbon to that high quality residue. And so we're going to have a nitrate depression period that is also called immobilization. So to improve microbial processes and nitrogen cycling and the synchrony of this nitrogen release, we need to take into account the residue quality that can be the type of crop, but also when it is terminated. The more lignin and the plant is going to be rigid and old, the higher the carbon to nitrogen ratio. And then all of those other things that Daniel talked about that are very important drivers of life. Temperature, pH, moisture, all of these are going to affect the ability of microbes to do their job and the soil food web as well. Starting soil organic matter levels, how much microbes do we have to start with? and what that community look like and some of the other management practices. Quickly, I'd like to show you a little bit of data here about how management could impact microbes. Really, it comes down to creating the resources and the habitat for growing microbes. Here you see total soil total soil carbon on the X, and on the Y-axis microbial biomass. And so the more carbon you're going to have in your soil, the more carbon additions you're going to have, the more microbial biomass you're going to have, and the more microbial activity you're going to have. So in general, if you have high degree of organic matter inputs and they are very varied, you have compost, cover crop, and some other forms of it, you will increase your mineralization and the amount of ammonia oxidizing bacteria that you will have. You also see a more connected microbial community and more cooperation between the fungi and the bacteria. As you increase fertility level and the amount of inorganic N in the soil, you will see a decrease in nitrogen fixation. And more bacterial dominance, especially those Pseudomonas, and those are the ones that are denitrifying, so some potential higher loss level. Eventually, feeding and protecting microbes through complementary plants, diverse rotation, keeping the soil cover, lowering the disturbances, and maintaining adequate moisture is going to be very important to provide the habitat for microbes to grow. What is coming up, though, in the literature is, do we want more of one specific bacteria or another specific bacteria? Can we steer that community? Can we engineer it the way we want? We're not there yet. However, what we know is that the more diverse is your microbiome and the more diverse is your food web, the higher level of nitrogen mineralization you're going to get. And the more nitrogen turnover you will have. Let me show you here for a second. Fostering biodiversity across that whole food web really tend to diversify the microbial function, so you get more microbes doing different things and even eating each other. You also have redundancy. You have microbes doing the same thing, and so if one of them die because it's more vulnerable to low soil moisture, some other parts of the community are going to take over and maintain that nitrogen cycle. So we do have redundancies between microbes, complementarities, and some that clearly depend on each other to grow and do the nitrogen and support the nitrogen cycle. Here is an experiment where they took a soil and they sieved it at various sizes of sieves to take out the fungi, take out the bacteria, take out the nematodes, take out the microbes, and incrementally remove some levels of organisms that are important to cycle nitrogen. And they looked at the impact of removing that biodiversity on mineralization and on nitrogen losses. And they found that as your soil, here you can see it on the x-axis, as your soil becomes simpler in terms of organism, you increase nitrous oxide emission. So you have a system that is imbalanced. As you increase, as you have a soil that becomes simpler, you also decrease your mineralization rates. So fostering diversity of microbes to the point of knowledge that we are at now is actually very important, is actually a better potential way to manage microbe than trying to steer your community one way or the others. So microbes are complicated. How can you take the pulse of what's going on? Still monitoring soil nitrate, and you'll go in depth of all of that later in the workshop, but monitoring soil nitrate is also important. But if you have low level of nitrate at planting, it can mean various things, or your microbes are very active and it's going to make it available over time, or it's not in, or there's not enough available. And so some things that you can do is look at your soil organic matter level. If it's high for your area, you could, and if you've been fostering that soil food well, it will likely be available over time. Or you can think of adding additional measurements of active carbon or microbial activity. We now have ways of looking at microbial activities that are like pH strips that just, it's a color indicator that tells you how alive is your soil and how active. So some key takeaway is that the soil biota is very important to cycle nitrogen, both in terms of releasing nitrogen from organic material into inorganic form, but also for fixing nitrogen and for those loss pathways. Our understanding of microbes hopefully is going to allow us to move from pools of nitrogen to flows of nitrogen. But really, when you think of organic nitrogen management, is really about understanding how it flows through that microbes, that food web. We're at the frontier of science. Only 10% of bacteria and fungi are known. So it becomes very difficult to be granular as to how to steer our communities. But we know that the more you diversify, the more biodiversity you have in your soil, the more likely you will have nitrogen transformation and reduce your nitrogen loss potential. And then finally, managing microbes really primarily involve manipulating the soil conditions. So trying to encourage their activity so that they can break down organic matter. And then adjusting this carbon to nitrogen ratio is also very important in providing microbes with the right diet and being able to see when that nitrogen is going to be available. With that, I thank you, and I'm happy to get onto some questions.
Great, thank you, Amelie. So we have 4 minutes for questions. So we'll do a few of them live together now, and then Amelie can visit the Q&A box and continue to answer them there. So one of the questions is, what happens to microbial populations in extremely hot environments? We get up to 80 Fahrenheit by February and over 100 by April.
Well, so it will be about temperature, but also moisture. So as Daniel has shown, when you increase temperature, you arrive at a sweet spot where microbial activity is going to be maximized. So you potentially have a very high level of microbial activity if moisture is adequate and if their food source is adequate as well. Yeah, now a soil is never dead. Okay, we can be in the middle of some desert valley. You'll find some microbes. So microbes are going to be there. They might be dormant, but they'll be there and ready to go as soon as the conditions become more favorable.
And the air temperatures are going to differ from soil temperatures too, right?
Yeah. And that will again depend on moisture. For a large amount, yeah.
How does microbially active containing compost, soilless media change nitrogen release? It would have higher levels of organic matter in the form of compost and peat. Would the same principles apply?
Sorry, that's a complex question. I'm trying to reread it. Can you read it again?
Towards the top of the Q&A. How does microbially active soilless media change nitrogen release? It would have higher levels of organic matter in the form of compost and peat. Would the same principles apply?
I guess so. I don't see why not, although it will depend of the microbial community and a whole bunch of factors that influence life and soil. But I think the principles should largely apply. They're based on ecological theories, and so now not all microbial community need the same thing either. And we're increasingly learning that some communities are more or less efficient at cycling resources, but we really don't know why, and so it's hard to actually forecast what would happen between different type of soils.
Okay, it has been said that fungal digestion is more likely to add to the soil organic matter pool, whereas bacterial digestion is more likely to provide plant available nitrogen. Do you agree with this?
Yeah, I mean that's some cool results that came out in the literature not long ago. That actually the necromass of microbes have different purpose within the soil, and the nitrogen that they release also have different pathways as to where it goes. And so that's why bacteria are super important. Actually, they're not the only organisms that are important for nitrogen cycling, but their favorite source of nitrogen producing organisms for plant uptake.
Thank you. I'm going to interrupt briefly, and Maria, if you want to post the poll, you can post the poll, and we'll ask one more question, a question about fungally dominated soils, and whether we want to be working towards fungally dominated soils over bacterially dominated soils, and which ratio is better for crop production, and is this actually true for annual crops?
Yeah, so this is a very difficult question, and I understand everyone is going to be frustrated with me because I'm going to be like, well, we don't really know. But what we do know is that in general, a fungal dominated soil, which doesn't mean there's no bacteria, right? You need both, okay? But when you have a higher level of fungi, it's going to be a soil that is more stable. So it will maintain this nitrogen cycle under varying level of soil moisture, which is important in irrigated agriculture, especially in those drip systems that have, high moisture around the soil, around the roots, and then not much around on the edges of the bed. So a fungal dominated system is going to be more stable, more resilient, and a little bit more balanced in working synergistically with bacteria. So you want both, but we often find that soils on organic farm and healthy soils have higher degrees of fungi. Now, more of that organic matter is going to be in the fungi biomass, and so it will really rely on creating the condition for that nitrogen to cycle from organic biomass of those fungi and bacteria into inorganic forms.
Great, thank you. There are so many really great questions in the Q&A.
I'll be working on it.
We'll try to work through those during the workshop, and maybe we can have a follow-up too by email. But we need to continue on. So, Maria, can you show the results from the poll for Amelie's poll?
So for the first question, yeah, it would be the lowest carbon to nitrogen ratio would be would provide the most nitrogen quickly. The bacteria that fix nitrogen is Rhizobium. And then the last one was a trick question. And so there's no right or wrong answer, but bacteria are important, but other part of the soil food web is important. So there's ambiguity here within the audience. There's ambiguity here in terms of the scientific community. And so I just wanted for everyone to understand that there's no simple answer when it comes down to soil life.
Thank you. Thank you for helping us embody that uncertainty as we figure this out. Okay, great. Daniel, go ahead.
Okay, great. Thank you. So in this presentation, I will focus on soil organic matter. And most of the research we have done was in fields. We took samples in spring, and these were fields with no recent history of manure application, of compost applications, or cover crops. So we were really interested in the soil organic matter. How much can it actually release during a year or during a growing season? And the questions we were interested in, here. So soil organic matter, if you have a soil with a soil organic matter content of 2%, there may be up to 10 tons of up to 2 tons of nitrogen in the top foot per acre. So there's a lot of nitrogen in the soil, but as we have seen, not all of it is available. But depending on how much is available, it can be a considerable amount for the crops. So in our research, we were interested in, first of all, which soil properties affect soil nitrogen mineralization rates of the soil organic matter. And the second question is, how much is actually mineralized every year and during the growing season? So if we do a budget calculation, what can we expect the soil organic matter itself to provide to the crop? The first question we addressed in lab incubations. We took undisturbed cores from many fields, kept them at the constant temperature for 10 weeks, and then determined how much nitrogen was made available by microorganisms under constant temperature, optimal moisture content. And the cores were collected basically in the northern half of California. Fresno sites were in the south. We went all the way up to Tule Lake, which is on the Oregon border. Most of the samples were from the Central Valley, but we had a few from Salinas. We measured a lot of soil properties and then did some statistical analysis to see which soil properties can best predict how much nitrogen was mineralized in these lab incubations. And we chose lab incubations because we could keep all the soils at the same, under the same conditions, same moisture, same temperature. Doing something like that in the field, of course, every field has different conditions, so that would be challenging. So in this graph, from left to right, that's the measured mineralization over these 10 weeks, and the vertical axis is what we predicted based on the soil properties we used. And we found that the best prediction is when we have a measure of the soil organic matter content, which makes a lot of sense because organic nitrogen is in the soil organic matter. So the more soil organic matter there is, the more can be mineralized. The quality of the soil organic matter also plays a role. We measured it in different ways. We did some physical separation to see how much is already highly decomposed, how much is still in the form of particulate organic material. We did some enzyme analysis to see how active are the microorganisms in the soil. So we found that a measure of the quality of the soil organic matter is also important. And the third factor is soil texture. Soil texture determines or has an effect on nitrogen mineralization. And that's kind of an interesting factor I'd like to briefly go into. So clay minerals, they can protect organic matter from decomposition. It may be physical protection. Let's say clay mineral help form aggregates, and inside of aggregates, soil organic matter can be protected from decomposition. It can be chemical direct interaction between clay minerals and organic matter, which makes it more challenging for microorganisms to decompose. So if we have identical conditions, then a clay-rich soil, clay-rich soils tend to have a higher soil organic matter content than sandy soils. And we saw that with our cores when we look at clay content from left to right and the total nitrogen in the soil. The units are gram per kilogram. That's equal to pounds per 1,000 pounds. Pounds of nitrogen per 1,000 pounds of soil. And you can see with increasing clay content, we also found in these soils from Northern California the total nitrogen, and with that the total soil organic matter content increased. And you know that too, when you have on your farm, when you have sandy soils and you have clay-rich soils, it's much, when you apply compost year after year, it's much easier to increase the soil organic matter content in a clay or loamy soil than in a sandy soil. But of course, when organic material is protected, that means when you have two soils, a sandy and a clay-rich soil that have the same organic matter content, then less of that organic matter, of the nitrogen in the organic matter, is mineralized in a clay-rich soil compared to a sandy soil. And in the same data set, we found that too. Again, the clay content and the y-axis shows the percent of the total N that was mineralized during these 10 weeks. And we clearly see it decreases. So on one hand, clay content increases the amount of soil organic matter and total nitrogen in the soil, but it decreases the percent of the soil organic matter that can be mineralized. And if we combine the two and also take differences in bulk density into consideration, we see that the effect is not as big. So here the y-axis is nitrogen mineralized in milligram per kilogram soil, or that would be pounds per 1 million of pounds. And again, the clay content, and you can see the effect is not that big, because on one hand, when we have clay, we have more, we tend to have more soil organic matter, but it's also less available. But so nitrogen mineralization depends on the amount of soil organic matter that's there, its quality, and then soil texture, especially the amount of clay plays a role, how much is available. So these are results from our lab studies, but when we want to know how much is actually released, then a lab study is not a really good way, because the conditions, of course, in the lab, in an incubator where we have constant moisture, constant temperature, are not the same as in a field. So when we ask ourselves how much is actually mineralized, we went to the field and did field trials. And what we did, in commercial fields, these were conventional fields, not organic fields, we had plots where the growers agreed not to apply any fertilizer. Then we measured how much is already available in the form of ammonium and nitrate at the beginning of the season. So how much nitrogen in the top 4 feet of the soil is already available, how much was added with irrigation water. And then at the end of the season, again, we took soil samples to 4 feet, analyzed for ammonium and nitrate. And we also analyzed the plant samples and determined how much nitrogen did the plants actually take up. And here's an example from one of the fields. So we consider input all the ammonium and nitrate that's already there in the soil in the top 4 feet, the first, second, third, and fourth foot. The field I'm showing here, we had a little accident. The grower applied some fertilizer N. It wasn't that much, but so there was some fertilizer input. Most of the other fields, we did not have a fertilizer input. Then some nitrogen was applied with irrigation water. There is always a little bit of deposition from the atmosphere. It is here in the fields we worked in, we estimated it to be less than 10 pounds per acre per year. And so then at the end of the season, we went in again, we measured how much of that nitrate was left in the soil and how much was in the plant. And we assumed that leaching didn't play a role because we measured all the way to 4 feet. So we assumed that nothing was leached below 4 feet. And what we found in this field is that at the end of the season, we had 80 pounds per acre more nitrogen than at the beginning of the season. And that the only input we didn't account for was nitrogen mineralization from soil organic matter. So in this field, the apparent nitrogen mineralization was around 80 pounds per acre. We did that in 13 fields across the Central Valley, and we found that on average about 75 pounds of nitrogen were mineralized during a nearly 140-day growing season in the top 2 feet of the profile. So we measured in 4 feet, but then also how much of that was actually mineralized in the top 2 feet, because most plants, the main root system is in the top 2 feet. So some roots go deeper, but they may not be very efficient at taking up nitrogen below 2 feet. when we had a look at how is that distributed during the year, we used the same curve I've already shown you that shows that during the winter, much less is mineralized than during the summer. So about 56%, a bit more than half of the total amount that is mineralized during the entire year was mineralized during the 139-day growing season during the summer, or about 1.19% of total N. So on average, annually, across all these fields, about 2% of the total nitrogen that was in the soil was mineralized during an entire year. And of course, it's a bit complicated if you want to determine how much is mineralized in your field to take all these factors into account. So for our worksheet, we present a much simplified version. You will find this table here that distinguishes between the Central Coast, the Sacramento Valley, and Imperial Valley. And for each of these regions, there are two different soils. One, for example, for the Central Coast and Sacramento Valley, 1.5% soil organic matter or 3% soil organic matter. So what you can do is to just add up when you know your growing season is from May to August, just add up the numbers. You find the soil or the column that best describes your soil, and you add up the numbers. These are values in pounds per acre to determine how much may be mineralized during the growing season from soil organic matter. So if your screen is big enough, we can do a little exercise using this table and determine how much nitrogen is mineralized from early April to late August in the Sacramento Valley soil with 1.5% soil organic matter. I'll give you a few seconds to determine if your screen is big enough that you can see these numbers. Otherwise, it's in the worksheet, the exact same table. So to determine how much is mineralized, you will find your columns, Sacramento Valley, 1.5% soil organic matter. You're interested in April, start of April until the end of August. So 4 plus 6 plus 7 plus 9 plus 8, that would end up being 34 pounds of nitrogen. And that's, I would say, a conservative estimate, especially for organic systems. We are now doing some research where we compare conventional to organic systems. I expect these numbers to be higher in organic systems, just as Amelie said, they are more active, the microbes are much more active, much more diverse. But as a conservative estimate, you can get you can use the table that's in your handout or in the worksheet. So to sum up, nitrogen mineralization from soil organic matter depends on how much organic matter is actually there, what is its quality and soil texture. And based on these trials in the field, on average about 2% or 2.1% of soil organic matter was mineralized per year in the top 2 feet in Central Valley soils under annual crops. And because some temperatures are warmer during the summer, the crops are irrigated here in California. Even though the growing season is may not be half a year, but about half of it is mineralized during a summer growing season. And as I mentioned, this is from conventional fields, and we are working now on organic sites to determine, is there a big difference between conventional and organic? Okay, thank you. And if you have questions, I'm happy to answer or try to answer.
Thank you, Daniel. Daniel, if you want to take a look at the results and tell us your thoughts.
Mm-hmm. So the largest pool of nitrogen in the soil, that's organic nitrogen. And nitrate and ammonium are much smaller, and ammonia even less. And the second question, the major forms of nitrogen taken up by plants, that's ammonium and nitrate. And the third questions, what do soil organisms do when organic substrates contain a lot of carbon but little nitrogen? They immobilize nitrogen. That's the term we usually use. So they incorporate nitrogen from the soil, from soil solution into the biomass, and that's what we call immobilization.
Great. Thank you. Great job, everybody. Okay, we're coming in for the home stretch. I'm going to go ahead and share my screen. Okay, so let's take a deep breath, everybody. Your heads might be beginning to burst, but here we are. So some of you may be thinking to yourself, Margaret, I thought we were going to be talking about compost and fertilizers and cover crops. We just spent all this time talking about microbes and what nitrogen is. And I want to highlight why we've done that and where we're going. So next week, we're going to get into the specifics of all of these materials that we typically think of as the nitrogen package. However, what you're going to find is that these materials are much more complex to estimate availability because of the nature of nitrogen and the microbially mediated process. And so this morning, Daniel highlighted how unique nitrogen is compared to our other nutrients. It's a nutrient that we need in huge quantities, and that is so important to getting good production. However, it stands apart from the others because of how dynamic of a nutrient it is. It can volatilize very easily. It leaches with water. And so our basic practices of how we incorporate it or don't incorporate it, how we irrigate, how microbially charged our soil is, all have tremendous impacts on that nitrogen source that we are adding to our soils. And then to really recognize how much of a factor the microbes are in these organically managed systems is so huge. Because, for example, let's take cover cropping. If someone does the same cover crop in the same field, but chops it down 3 weeks earlier than the second half of the field, the timing of those of that nitrogen, the amount of nitrogen, etc., is going to have a big difference because we're relying on microbes. The temperature of the soil is going to be different. The C to N ratio of a cover crop 3 weeks later is going to be different, and the total nitrogen is going to be different. And so these very small differences in the compost we're using, the amendments we're applying, the residue that's being incorporated are having tremendous impacts on nitrogen management. And so we take time to really emphasize this microbial process and how big of a factor it's having on our organic nitrogen dynamics. And next week, when we talk about compost and fertilizers and cover crops, it will become even more evident. But this background will prepare you for understanding those dynamics. I want to share with you this figure, which is a model that we created to show this all the contributions of nitrogen in an organic operation. And so this bottom band in the sort of darker brown, or in the brown, that's the soil. So that's what Daniel Geisler was just describing. That's the soil organic matter contribution. And it's surprisingly significant. I always found it very significant or surprising because it's kind of this silent contributor in the background, releasing really tremendous amounts of nitrogen in the system in this sort of slow release way over the season. That next band in green is the contributions from cover crops. And it's also a very significant contributor in a single season. So here we have April through September. So this is modeling an example of a summer growing season. That next band is the blue band, which represents the contribution of compost to the plant available nitrogen that summer. And then the following band in purple is the granular fertilizer. In this case, we did like a 4-4-2 type material. And lastly, the irrigation water. And so by the end of next week, we will have gone through each of those bands, except for irrigation water. We do that on the last day, and begin to understand how collectively these inputs are all generating the nitrogen supply needed for the crop demand. So hang tight. Next week will be the next piece of the puzzle. And so today I want to end with establishing nitrogen demand. And so we're here talking about nitrogen, and it's hard to know how you're doing or what to do if you don't have a goal. So I just want to touch on that to give everybody some tools on how to establish that goal. So in our worksheet, this is section 3. This is your nitrogen demand in part A. So you can reference that. When we talk about nitrogen demand, this is the nitrogen required to produce the whole plant. It includes all the leaf, tissue, and the stems, the fruit that you're going to harvest, and any of the culls that got left behind. And although it is including all of that, the way that it's usually estimated now is to connect it with yield. And so when you look at estimates of nitrogen demand, it's important to know that that's connected with yield. And so you can make adjustments based on your anticipated or known yield. So here's a table that is in the 8712 document. And it gives you some examples of yield. And it's important to know that in this resource, and likely many others that you might look for these estimates, it's important to be tracking what those numbers, how those numbers were achieved. And typically they are numbers that are from conventional operations, that are from commercial growers that are growing these crops in the optimal environments. And so they tend to be higher. But it's important to use that and then adjust it based on your own knowledge or best guess. And what we also have here in this table, so the first column you have the crop, the second column we have the example yield, so we can just go through lettuce. So 20 tons per acre. However, this third column is neat because it gives you the pounds of nitrogen per ton yield. And so in this case, we're going to take 20 times 8 to get 160 pounds of nitrogen per acre. That's going to be the total crop N uptake. And so you can adjust that total N uptake based on your yield by using this multiplier here. So let's say, for example, you only expect 10 tons to the acre of lettuce. You can multiply 10 times 8 and have a crop and uptake of 80 pounds to the acre. And so here's a starting place to help you establish your crop demand. Here's another resource, and you can see like a range that's provided. So take some time to figure out what your goal is and how much you think your crop needs based on your yield, your environment, your expectations. So another factor to consider is that while some crops can be grown in both winter and summer, they will usually take up less N in the winter planting. So there can be some adjustments there. And another consideration is that because plants do not capture all the nitrogen that's applied, the amount of total fertilizer nitrogen needed for the optimal growth is often a little more than the actual crop uptake. And why might this be? Couple of reasons. We have some inefficiencies in irrigation management. And so this is going to impact the timing of demand and supply, and also the location of that nitrogen and the depth of the roots. And a second factor is the natural variability in a field. And so as you'll see through this whole workshop, we're really trying to master the art of making our best guesses. And as growers or advisors, taking into account these inefficiencies and variabilities that exist. Another factor is the influence on soil texture. And this can happen within a single region, or if you're working across regions, you may even have greater variability. And so the leaching of residual nitrate is more pronounced in sandy soils than clay soils, which we've covered a little bit already. So for example, on a sandy soil following a wet winter, it's often necessary to apply more fertilizer to make up for that loss that happened with that water. So although you want to have a goal for your nitrogen demand, it's important to maintain that this is a starting point and not a prescription. Here are some additional places to find some of those demand numbers. And I can drop this, I'll include this link in the follow-up email. So this is the California Fertilization Guidelines. And you can find them for these crops. And what you'll find in some of these is a page that looks like this. So you can get the timing of the uptake. You can get a sense of how much of that total N gets used to invest in the vines, the material, the plant tissue, versus the fruit versus the culls. And then you can get some total demand numbers. Let's see. So that's my wrap up for the nitrogen demand section. And so to touch on next week, we will be meeting on Monday from 1 to 3. And next week, we will be covering compost, fertilizers, cover crops, and crop residue. And Patricia and I will be here to go over those materials. And if you want to prepare for next week, you can read the next 5 pages in that 8712 document. And then in part 3 is when we will be tying all of these materials together. And so we'll look at how to complete a nitrogen budget, and to get those pieces of soil organic matter, cover crops, compost, fertilizers, to estimate those numbers, and then put them together with the crop demand and the anticipated release. And then we'll also bring in how to use soil and tissue tests, and how to consider nitrogen that's coming in from your water. And then the last 30 minutes of that session, we have a kind of fun, cutting edge science portion of our workshop that we look at some of the frontiers in nitrogen science. So what's some of the cutting edge information that's maybe where we're going to be going with some of our thinking and explorations in nitrogen science, especially as it pertains to organically managed systems. And then part 4, we'll have a grower panel, and we'll have an opportunity to have some lively discussion with some practitioners who have thought a lot about nitrogen, and it's a great chance to have a meaningful back and forth, both with the organizers of the workshop, the grower panelists, and the participants, all of you. So stay tuned. Let's see, any questions before we end for the day? We have a few more minutes. If I can address any maybe logistical questions, or if Amelie or Daniel or others have any thing to share or add.
There's one in the Q&A for you, Margaret, that asks, are there any options for crops where yield is not measured in weight? In my case, it's cut flowers. Thank you.
Well, that's a very good question. So I haven't looked into flowers myself, but I expect that there are estimates for nitrogen demand for flowers. And the removal portion, we'll talk about a little more next time, because it affects the crop residue, about what's going into the field after harvest. So again, flowers will have its own proportion of the crop that is removed with harvest versus being returned into the soil. But I don't know if you've, whoever asked the question has found nitrogen uptake demands, but I expect that flower crops have their own series of demand uptake numbers that have been established by the industry. And I think similar adjustments would be made based on your expected yield, based in bundles, or whatever the unit of sales is for that crop, and can be adjusted to your own local sales. Okay, great. Well, Amelie and Daniel, thank you so much. And Maria, thank you for your support. And we will continue to, we'll address any unanswered questions as we can, and hope to keep up with those until next week. I will send a follow-up email in the next few days that will allow you access to the recordings and any other information that we have. So thank you all. Have a great rest of your Monday. And I'll see you after the big storm. I hope the rain comes, at least for us around here. So take care, everybody. And thank you for being here.